We're going to move on to very exciting hot topics in multiple myeloma. The first speaker will be Dr. Ken Anderson, talking about minimal residual disease.
Quick question. Could I announce the winner?
Thank you very much. I do want to thank Pat for sharing that very heartwarming and emotional story. I think all of us can identify with it. And I do think that there's a silver lining in this myeloma diagnosis for the patient and their family, and it is really to appreciate the small things and the important things in life even more than we perhaps wouldn't have otherwise. So thank you very, very much. I do want to, as Leif mentioned, just talk about minimal residual disease, and this is something that I think you've heard about previously, but I'll just start off by saying that minimal residual disease means, in fact, small amounts of multiple myeloma, at least for us. And you've seen this slide previously. The top of the iceberg is what we used to be able to see in myeloma, but with more sensitive techniques, we can detect what we call deep responses under the water here. So we can detect fewer and fewer numbers of cells. And in fact, a complete response, as we have called it over the years, which means fewer than 5% plasma cells, unfortunately still could mean up as many as 100,000 cells still in the bone marrow. So what you've heard today at this wonderful meeting is we have new drugs that we use 3 of them together, with or without a transplant, and we can get an unprecedented level of response. So we really do need to measure at the bottom of this iceberg. So what is minimal residual disease? It's one myeloma cell in 100,000 normal bone marrow cells. And someday, pretty soon, I think we're going to change the definition to one myeloma cell in 1 million marrow cells. But the point is, this is a level of depletion we have not been able to appreciate together before. And now that we can, because of the novel immune therapies you've heard about here today, together as a community, we need to figure out what to do with it. What does it mean clinically, and how can we use it to help us develop and get new drugs approved even more quickly? So MRD positive, when you hear this and in talks or you see it in papers or whatever, MRD positive means there's still tumor cells there, not that you would see under a microscope, not that you may measure because of protein in the blood and or urine, but because by very sensitive techniques, there's still one myeloma cell in 100,000 normal cells. Conversely, if you can't find that very few number of cells, we call it MRD negativity. So what's been done now in the last couple of years is to determine what is it that's so important about MRD negativity. Should we try to have a goal in every single treatment to make somebody MRD negative? Can we inform treatment? Should we use something more aggressive if you're not MRD negative? Or the one I like the most is in terms of maintenance. If you're on maintenance therapy and you become MRD negative, could we potentially stop the maintenance? All those are questions that need to be and are being answered now in clinical protocols. This is an example of why this is relevant. This actual curve here is all the patients who are in complete response by conventional measures. This happens to be the trial that the Dana-Farber did together with the French of RVD with or without transplant. It really doesn't matter. The point of the slide is all of these people are in complete response. And they've then been measured for minimal residual disease. And you can see that the people who don't have minimal residual disease are doing better than the patients whose disease is coming back more often who do have MRD positivity. And this was at the level of 1 in 100,000. So what has been done, and this is my colleague Nikhil Munshi and a number of us together, is we actually looked at all of the literature that's already published, and there were 14 different studies, lots of patients, and all of them looked in particular at all of them, of the people who had a complete response by standard measures, proteins in the blood and or urine, And could you divide them up into MRD positive and negative? And if you could, did it make any difference? So what Nikhil published about a year ago now is looking at MRD in these studies by 2 different ways and asking whether it made a difference. So the first way is flow cytometry, and I'll tell you about that in a minute. But basically, think of it this way. It's fingerprinting the myeloma, looking for 8 different proteins on the cell surface usually, and each one is bound to a different color. That's how we do it. Or, as you'll hear about from Jonathan with elegant gene studies, figuring out and counting the myeloma cells burden by looking at the genes that are there that we know characterize myeloma, not normal cells. And what Nikhil showed was that in the patients who had a complete response, by conventional measures, both the progression-free survival, that means the time without active myeloma, and the overall survival, which means how long you live, were better if patients got MRD negative. So within the complete response category, it didn't matter to go to the extra level and measure that level of depletion because it seems to correlate in the previously published trials with outcome. So here are the 2 ways to do it. Flow cytometry is the fingerprinting I was talking about, staining the myeloma cells quite literally for 8 different proteins, using different colored dyes, and finding as few as 1 in 100,000. The advantage of that is you can use it for every single sample. You don't have to have a sample from earlier, maybe at the time of diagnosis, so that's all good, and it's widely available. The gene sequencing is really very, very sensitive. I think it's going to be the way we go in the future, but suffice it to say, you do need to have some sequencing done from prior times when you actually got diagnosis so we can know what the sequence looks like for your particular multiple myeloma. And you can see the disadvantages are on the other side. For the fingerprinting, we need to use fresh samples. Those aren't always available. And I already mentioned for the VDJ or gene sequencing, we really need to have an early sample in order to understand what we're looking for later on in your disease course. So here's a slide again from our French-American, I call it the DFCI-IFM trial, but it tells you that it's probably better to get rid of more cells. So this is like different levels of depletion. So if you go from the bottom to the top, at the top line, you're down to fewer than 1 myeloma cell in 1 million normal cells. And you can see that those patients have the best outcome versus those that have 1 in 100,000 or 1 in 10,000 or even 1 in 1,000 as it's going down lower and lower on those curves. So right now, as I've already said, we look for minimal residual disease in the bones, in the bone marrow, using one of those 2 techniques or both. But there's also imaging that all of you have undergone, MRI, but in particular PET-CT scanning, because we want to be able to look not only in the bone marrow, but outside the bone marrow for small numbers of cells. And so Shaji Kumar from the Mayo Clinic has just actually published MRD negativity, which means fewer than 1 in 100,000 cells being myeloma in the marrow, in association with a negative scan that's called an imaging MRD negative state. In the future, what's going to be done, I would predict for you, is we're going to look in the blood, because as Jonathan may mention, we can sequence as few as 1 myeloma cell in the peripheral blood, or we can even look at what's called cell-free DNA. That means the genes from the myeloma that are circulating in the liquid part of your blood, even if there aren't any myeloma cells there. So one or both of those, or maybe together, will be used, I think, in the future and monitor your disease and the different clones, as Leif has talked about over the years, in the blood without potentially having to do a marrow. That remains to be seen. So here is just a couple final thoughts. I've already said it, with drugs with or without transplant, we can achieve MRD negativity, so we as a community need to understand what it means. It looks like it's worth achieving because people have a pretty good and a better clinical outcome if they achieve it. The technologies to measure it, I've already called flow cytometry and gene sequencing. But the unanswered questions are two kinds, and I just want to stress this really importantly. Clinically, we really don't know right here whether we need to change treatment. So if you get on initial treatment and you're not MRD negative, should we switch the drugs? If you get a transplant and you're not MRD negative, should you get some consolidation therapy? Or the third example is if you're on maintenance treatment and you become MRD negative, can we stop, which we don't do right now. The other thing I want to mention, which isn't on the slide, and it's really important, you know, this meeting is all about all the success we've made together in developing new drugs. It's been awesome, but if that success is going to continue, we need to have MRD or some other surrogate marker. The plain truth is, if somebody gets new myeloma today and they get on standard drugs with or without a transplant, their progression-free survival, the time without myeloma, is at least 7 to 10 years, if not longer, with maintenance. So if you were a drug company, and there are some of them here, and you had a new drug that you wanted to get approved in an early initial treatment of myeloma, and you wanted to make that another 30% or 50% better, so if it's 10 years, so that would be 15 years of progression-free survival time without myeloma. Drug companies can't wait that long. It's way too expensive. And most importantly, we as caregivers, but really importantly, you as patients cannot wait that long for a new drug. So we're very involved at the level of the foundation of the NIH, the FDA, and others. What's ongoing, all of the investigators here, we're pooling 3,000 patients' data, 15 different clinical trials, including drug company trials and also cooperative groups and different centers. And we're going to correlate MRD status with clinical outcome. Does it matter how you get to MRD with or without a transplant? Is an MRD in somebody who has 414 translocation the same as MRD who has 17p deletion or hyperdiploidy? These are questions that all need to be answered. But my prediction for you is within the next year or two, the FDA is going to make the jump. And they're going to say that you can use MRD as a regulatory endpoint, and I'll just make up, say, at a year and a half, in terms of MRD negativity being 50% different or a third different, rather than waiting for progression-free survival, which wouldn't occur until 10 years. And that's a big deal because that's how you and I and all of us together are going to get new drugs that you all and you're going to need and we're going to want to have to help you in the years to come. So I think I'll stop here. Thanks.